and go.
Hello, everyone. Welcome to today's live broadcast, POLYOX, Polyethylene Oxide Essentials, an expert guide to polymer properties and drug formulation. I'm Megan Manzano, and I'll be your moderator for today's event. We are pleased to bring you this webcast presented by Pharmaceutical Technology and sponsored by IFF. I would like to share a statement from our sponsor before we begin. IFF Pharma Solutions pioneered excipients like Athocel and Methocel more than 75 years ago, and they have since created a unique range of well-known excipients designed to deliver performance and cost advantages across oral dosage, oral liquids, and topicals. Their commitment to quality, service, and reliability isn't to check a box, but to drive an endless pursuit of delivering the right answers at the right time. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found at the bottom of the video player. You can enlarge the slide window by clicking on the small icon in the bottom right corner of the media player. The slides will advance automatically during the event. If you have any technical problems viewing or hearing this presentation, please click on the question mark help widget in the top right of your presentation window. I would now like to introduce today's speaker. We are pleased to be joined today by Elizabeth Tochi. Elizabeth is a lead application scientist for IFF Pharma Solutions, where she heads technical marketing projects promoting the expertise of the Pharma Solutions business unit. Elizabeth joined the IFF Pharma Solutions team in 2018 as a pharma application development scientist. In this role, she provided technical support to pharmaceutical customers and project leadership on growth and innovation projects using IFF's pharmaceutical excipients. Prior to her roles with IFF, Elizabeth was a research and development scientist for the Dow Chemical Company's Pharma and Food Solutions excipient business, where she led R&D projects in areas of specialty oral solid dosages and quality improvement. Thank you so much today for joining us today, Elizabeth. Please get us started.
Thank you. Thank you for having me. Let me share my screen. Okay. So today is about POLYOX, polyethylene oxide essentials. When I thought about this title again, it kind of sounded a little bit like a class or a university course. So I thought we'd start off with a quiz. What do paper towels, razors, osmotic pump tablets, and oral thin films have in common? Polyethylene oxide. POLYOX, polyethylene oxide, is a polymer with a wide range of uses, most of which depend on the high affinity it has for water and the different characteristics that it has when it is hydrated. I will not go into home and personal care applications for POLYOX, as that would be its own webinar. For simplicity, I'm going to take my video off so you can see the whole slides. Okay. All right, I think we're back. What I will cover today is an intro to POLYOX, polyethylene oxide, material properties, its uses in pharma applications, and I will also spend time on an important property of POLYOX, which is autooxidation. We will have time to take some questions at the end. POLYOX is a trade name for polyethylene oxide products. Polyethylene oxide, or PEO, is a non-ionic hydrophilic polymer that can be used in modified release and specialty matrices. It is a solid white powder that is differentiated from other hydrophilic polymers by its swelling ability, mucoadhesion, and mechanical strength. POLYOX PEO grades differ based on viscosity, particle size, and residual ethylene oxide. I'll give a brief history of POLYOX, or polyethylene oxide. So PEO was first discovered inside an ethylene oxide cylinder back in 1951. In less than 10 years, the team developed a catalyst and a small scale production of polyethylene oxide. It took another 10 years to build a large-scale production facility, which is still standing today. Almost 20 years later, in the 1980s, ALZA developed the first oral osmotic pump technology using POLYOX PEO, which initiated the need to make a pharma grade of POLYOX. Pharma grade POLYOX PEO was introduced to the market in the 1990s. There have been many great additions to the POLYOX PEO family over the years. Next year, we will introduce a new packaging to extend the usable life of Polyux. And I'll touch a little bit on this later in the presentation. Polyethylene oxide is chemically identical to polyethylene glycol, with the difference coming in the molecular weight ranges of each. PEG, or polyethylene glycol, is defined as polymers less than 20,000 Daltons, and PEO are greater than 20,000 Daltons. The chemical synthesis is slightly different, which allows each process to reach the desired molecular weight range for each product line. Polyux uses a proprietary catalyst system to synthesize very long molecular weight chains. Through controlled chain decision processes, the molecular weight is reduced to the targeted molecular weight for each grade. I like to think of this as spaghetti. We make really long spaghetti strands at first, and then your toddler doesn't want to eat or twirl the long spaghetti strands, so we go in and cut down that spaghetti to the desired length so that they can eat it easily with a fork or their hands. That's kind of what we're doing here with Polyux. The nomenclature of Polyux is outlined on this slide. We begin with Century, which is the trade name for regulated grades of Polyux PEO. Then we have the trademark Polyux for polyethylene oxide, WSR, which stands for water soluble resin. We have a viscosity indicator, and I say indicator because there's no correlation between the name or numbers listed here and the target viscosity or molecular weight of the grade. There could be additional indicators such as LEO for low ethylene oxide, which means no more than 1 ppm residual ethylene oxide, or FP for fine particle grade, or SFP for superfine particle grade, and lastly, NF, which states that it meets national formulary compliance. This table shows the available polyethylene oxide grades for pharma. The main distinguishing factor is the target average molecular weight, measured using solution viscosity as the specification. The molecular weight ranges from 100,000 up to 7 million Daltons. Due to the viscous nature of PEO, the solution viscosities have to be measured at different amounts. For the lower molecular weights, that's 5%. For the higher molecular weights, that can either be 2 or 1 weight percent. A subset of these products are offered in fine particle and superfine particle grades. Let's talk a little bit about the properties of PEO and applications for PEO in the pharma space. Polyux has many different properties that make the polymer useful in pharmaceutical applications. It's hydrophilic, pH stable, thermoplastic, mucoadhesive, non-ionic, and viscous. Over the next several slides, I'll highlight how these characteristics or Polyux are used in pharma. These are the typical applications that polyethylene oxide are used in. They're mainly specialty oral matrices, including controlled release matrices, osmotic pump tablets, buccal or oral tablets and films, gastroretentive tablets, and abuse deterrents. For controlled release matrices, the molecular weight of the PEO polymer impacts how an active will release from a PEO matrix. So the molecular weight or the viscosity is impacting controlling the release rate of the active from the matrix. As the molecular weight increases, entanglements increase and increase the gel strength, slowing down the release of the active. For water soluble API, the diffusion pathway lengthens. For poorly water soluble API, the disintegration time lengthens, thus slowing the API release. The example on the left shows caffeine as a sparingly soluble model compound. The release is fastest with the low molecular weight, 100,000 Dalton, and it slows considerably with the two highest molecular weights of 5 million and 7 million Dalton, shown here in blue and green. The less soluble phenacetin on the right, it also shows the same response, a similar response to molecular weight, with a very slow release rate of the active using a very fast release rate using our low molecular weight N80 or the 200,000 Dalton grade. And that release rate slows as you increase the molecular weight to a coag, which is 5 million. Um, and this is due to the very slow disintegration time that this grade has in water. POLYOX, polyethylene oxide, makes a good choice for matrix tablets because its solubility is stable over a wide range of pH that include those found in the gastrointestinal tract. The graph on the left shows the precipitation temperature of PEO in water at different pHs from 1 to 13. So the temperature is the precipitation temperature is relatively stable around 95 to 100 degrees Celsius when the pH is less than about 11. You can see it's very stable. When it reaches around 12 or 13, you see this drop off in the precipitation temperature, indicating that the polymer is crashing out of solution. The right graph shows a model matrix formulation using theophylline and POLYOX PEO. The tablets were put into 3 different pH mediums ranging from 1.1 to 6. The dissolution profiles are almost identical, showing that the release from PEO tablets is pH independent as long as the API solubility is pH independent as well, or not sensitive to pH changes. All right. One of the most distinguishing features of POLYOX or PEO is the swelling of the polymer. PEO hydrates very quickly and, depending on the molecular weight, swells to many times its original weight. The swelling graph in the center shows pure polymer compacts of both POLYOX and Methocel HPMC. These compacts have been put into 900 milliliters of water, and the weight gain measured over a 24-hour period. These compacts were allowed to erode and dissolve into the medium. The solid lines show POLYOX PEO. Green here is the highest molecular weight we have for pharma of 303, and red is another high molecular weight, 301, of 4 million. And you can see that their swelling is very rapid, and they continue to absorb water even at the 24-hour mark. The mid molecular weight PEO down here in the blue is a 1 million molecular weight polymer. We can see the initial rapid hydration similar to the high molecular weight polymers, but this plateau and then decrease in the weight gain at up to the 24-hour mark. This is happening because it's eroding into the water, and it's fully eroded at the 24-hour mark. In comparison, we have high molecular weight Methocel HPMC shown in the dashed lines. These show about half as much weight gain as we get with the high molecular weight POLYOX PEO. The green dash line is green dash is Methocel K100M. And this is one of the highest molecular weight HPMC polymers we offer. The red line is K4M, which is the same substitution chemistry as the K100M, but just a slightly smaller molecular weight. In blue is E4M, which is a slightly different substitution chemistry from the K chemistry, but similar molecular weight as the K4M. And you can see all 3 do not perform as well as the POLYOX PEO polymers perform. POLYOX high swelling property is used in many specialty applications, including osmotic pump tablets, gastroretentive tablets, and abuse deterrents. I'm not going to go too much into the gastroretentive application. Essentially, you use the high swelling property of PO to prevent it from progressing through and past the stomach. And it's a lot more complicated than that, but for simplicity's sakes, that's what's going on. Let's talk about osmotic pumps because this is the main application space for POLYOX, polyethylene oxide. Um, there are many benefits to osmotic pump tablets, and that are listed here. They um, they allow for controlled release up to 24 hours. They are good. They have good in vitro in vivo correlation, steady state or customizable plasma levels through zero order or again customizable release profiles, and they are resistant to food effects. There are different kinds of osmotic pump tablets. Some that you can have a monolithic core where you just have essentially a matrix covered by a semi-permeable membrane, and then either a laser drilled hole or pores that are in that semi-permeable membrane. You can also have multi-layer cores where again you maybe have a push layer or drug layers and that are covered by a semi-permeable membrane, again with either a laser, most likely a laser drilled hole, and which allows for migration of the drug only through the holes that are drilled into it. The video shown here is of two tablets. One tablet that we made in our lab on the left, and compared to a tablet that we got off the market. So both contain the same active ingredient, trying to replicate the innovator. What I want to show here is how the osmotic pump tablets work. This has an aesthetic coating that surrounds the semi-permeable membrane. So this is put into water. You can see the aesthetic membrane starts to come off. That pink goes away, and the cellulose acetate coating is clear. And you can see the two different layers of this osmotic pump. In this case, we have a push layer in red and a drug layer in yellow, with probably hard to see, but there is a drilled hole at the top of this to allow. So what the semi-permeable membrane does is allow water in, but no drug out. What POLYOX is doing in the push layer is swelling and pushing on that drug layer to start pushing out. There we go. You can start seeing the yellow coming out, pushing that drug out of the drilled orifice. And we can, as you see over time, this red layer is expanding as it absorbs more water and pushes out that drug until you get pretty much complete drug release, and this whole core is red from the push layer that contains a high molecular weight polyethylene oxide. So I'd mentioned there's a lot of good benefits of osmotic pumps. There are some downsides of osmotic pumps as well. This includes they're quite expensive to make. They have a long manufacturing process because you're creating tablets, you're coating them, you're drilling holes. It's much longer than say your average matrix tablet. They do have a limit to how much drug can be put into them because you can't create a massive tablet that someone can't swallow easily. All right. How does PEO and osmotic compare to other materials? This slide shows commercially available osmotic pumps that use PEO in the push layer, a mixture of PVP, HPMC, and SSG in the push layer in that center one, and alginate in this bottom one. So what we did is looked at the the how much drug came out of each of these commercially available tablets. And we could see, and again, the push layer is red, the drug layer is yellow. We could see that after I think we did a 24-hour time period, almost all the drug in the PEO containing tablet is pushed out. There's some remaining in both of the other tablets. Quantifying the amount of drug that remains, we see that PO has anywhere from around 6 to 8, depending on, it looks like a little bit difference in the pH environment, but not by much. And the PVP, or sorry, yeah, the PVP, HPMC, and SSG and alginates had almost double the amount of drug remaining in the system, around 12 to 16%. So PEO systems are possibly more efficient in getting out drug in an osmotic pump tablet than other polymer systems. All right. One of the last properties, second to last property I'm going to talk about with POLYOX is its mucoadhesive property. This makes it so when it's wet, it becomes mucoadhesive. When it's dry, it's not mucoadhesive, but when you wet it, it becomes mucoadhesive, and your mouth's wet. So you can easily place a buccal tablet into the oral cavity, and it'll stick there for long periods of time. Same thing with films. You can stick it into the mouth, and it'll stay there. And depending on the molecular weight, you can have controlled or immediate release, depending on what it is that's desired outcome or performance from the tablet or film. The data on the graph shown here is adhesion levels of different grades of POLYOX PEO using two different methods, an in vitro method in black and an in vivo method in data shown in gray. This is an article that was published back in 1991. The article was partially highlighted the drawbacks of using an in vitro test method and showing the benefits of using an in vivo test method. So I'm going to focus on the in vivo data because that's more relevant than an in vitro data. The in vivo data shows an increase in mucoadhesion as the molecular weight of PEO increases from 200,000 to 5 million. Around 400,000, there seems to be a plateau where additional molecular weight, increasing the molecular weight is not imparting significantly higher adhesion properties. The last property I'm going to talk about before we move on to discussing autooxidation of POLYOX is the thermoplastic nature of PEO. So PEO films. So PEO is thermoplastic. Its melting point is around 70 degrees Celsius. At this temperature, the material softens and extrudes very nicely, especially with the lower molecular weight PEOs. The thermoplastic nature of the polymer, along with the fact that it's inherently tough, makes it ideal for abuse deterrence. In this case, we're talking about thermally curing a tablet of PEO, which essentially fuses the tiny tough particles together into one tough large particle or tablet that's hard to crush and hard to grind. Pairing that hard to crush and hard to grind with the fact that a high molecular weight PEO swells significantly makes it advantageous in abuse deterrence in deterring both crushing into a powder for sniffing up the nose, or crushing to a powder, putting into water to hydrate, and then extracting the active using a syringe and trying to inject it. It becomes really thick and viscous, and it's very difficult to take up with a syringe and to then inject into the body. So those two properties are why it has been historically used in abuse deterrence. And lastly, again, the thermoplastic nature means that it makes a really nice film. Now I want to move on to auto-oxidation of a PEO. So PEO undergoes auto-oxidation. There are several pathways that it can go through auto-oxidation. I'm just showing one potential pathway using a free radical where you're essentially causing chain scission of the chains. And then that chain scission reduces the molecular weight, which reduces the effective observed solution viscosity of the material. There are many factors that impact the auto-oxidation rate. The auto-oxidation can be catalyzed by higher temperatures, exposure to UV light, certain metals can catalyze auto-oxidation, pH impacts it, crystallinity, and percent relative humidity. Why should we care about auto-oxidation? Well, it happens it happens right when we start right after we make it. And when it's sitting in storage, this is happening. So if from the time that we make it, the molecular weight may be significantly different a year later, and we need to understand how fast is it degrading, how is that potentially impacting applications and performance, how do we control, and then different ways how can we control and slow down the rate of auto oxidation. All right. Auto-oxidation is random chain scission. We're not talking about cleaving at the end of a chain and creating tiny little individual monomers. We're talking about randomly cutting the chain somewhere along the chain, and without truly understanding exactly where. It's going to just be random. So what I'm showing here is that the chain scission rate is going to impact the observed solution viscosity of higher molecular weight chains more so than lower molecular weight chains. The example I'm kind of using to display this is N60K, which has a molecular weight of 200 million. It has 4 chain scissions, it's cutting it down to 5 chains of average molecular weight of 400,000 per chain. That's an 80% reduction in the molecular weight from a starting of 2 million. In comparison, I'm showing N12K, which is approximately 1 million molecular weight chain, but we have to do it on the same weight basis as the N60K. So 2 chains of N12K equal the same, mass as the N60K. So same 4 cuts over 2 chains of N12K result in 1, 2, 3, 4, 5, 6 new chains with average molecular weight of 330,000 daltons. That's a 66% reduced loss in molecular weight. So you can see how it's going to that loss impacts the higher molecular weight more so than the lower molecular weight. And the chain scission rate for that we've been able to understand is independent of molecular weight for the most part. And so this is the assumption that that you will see similar chain scissions per mass between the different grades. All right. This impact on the difference in chain scission rate, the similar chain scission rate, but the difference in the impact on observed solution viscosity impacts our retest intervals for polyethylene oxide. For our lowest molecular weights, N80 and N10, we have a 2-year retest interval. It doesn't affect the viscosity as much as these higher molecular weights. We can have a longer retest interval. For the bulk of the middle grades, we have a 1-year retest interval. And for our very highest molecular weights, coag and 301 or 303, we only have a 6-month retest interval. As I mentioned before, we will be launching a new package type to extend the retest intervals of everything to 2 years in that new packaging. Um, but that's to come next year. I'll highlight a little bit of data in the next couple of slides or in a few slides on how that packaging is going to allow us to extend retest intervals for PEO in that packaging. All right. All right. I mentioned again molecular weight impact of autooxidation. Higher molecular weight has observed faster viscosity degradation over time. Here I'm showing a low molecular weight and 80 200,000 molecular weight in this blue line. This is stored at ambient conditions for up to 12. This is 12 months. You can see a slow decrease in the observed viscosity over that time. In comparison, one of our highest molecular weights, 303, 7 million Dalton, we see a more rapid decrease in viscosity over that same 12 months. We add BHT to help slow autooxidation. BHT is a free radical scavenger that is added to every batch of POLYOX polyethylene oxide. This data is N80 data looking at how the BHT impacts the viscosity stabilization, how it stabilizes the viscosity over time. The blue line is showing a batch of PO where 500 ppm BHT was added to it and then put into storage. The pink is showing where no BHT was added and then put into ambient storage. You can see the drastic difference, how the no BHT, the viscosity loss is much faster over the same time as adding the 500 ppm BHT. We add BHT to help stabilize the product. Other factors that impact the observed viscosity decay rates of POLYOX PEO are temperature and humidity. So we'll start with humidity. On the left, we put small amounts of PO into baggies. We kept the temperature constant at 25C, and then raised the relative humidity from 35% to 60 to 75%. So these samples at 25C with 35% and 60% relative humidity conditions showed pretty good stability over the 40-day time period that we were measuring. When we raised that relative humidity to 75%, we saw a drastic decrease in, or increase in the viscosity loss over that same time. In the same study, we were testing the impact of temperature on the viscosity loss. So these are two different grades of PEO. We have the high molecular weight 303 up here in red, and still a high molecular weight, but just the one grade lower coag here in the dark blue or black lines. With the diamond shapes, those were stored at 25C, 75% relative humidity, not a great choice for relative humidity, but we kept relative humidity constant for all of these samples. So 25C in diamonds, we have 40C in the circles, and we can see how just raising temperature from 25 to 40C, we see a drastic drop in the viscosity over time. Same thing for the coag. Diamonds are 25C, circles are 40C. We see an increase in that viscosity loss over time. So increased humidity, not good. Increased temperature, not good. How can we help reduce the autooxidation rate of PEO? I already showed that we add BHT to help reduce that rate. Storage conditions can help reduce the rate. So the first thing that I always recommend is to store as cool and dry as absolutely possible. This data is showing the viscosity loss of PEO 303, POLYOX WSR 303 over a 24 months time period. We stored commercial batches, this batch in a drum in refrigeration in the blue, another drum in a controlled environment of 25°C, 60% relative humidity, and then the third in accelerated conditions of 40°C, 75% relative humidity. So you can see how refrigeration really helps slow down that auto oxidation rate and retain that viscosity for much longer than more ambient like conditions, and definitely much better than accelerated conditions. I mean, I can't stress enough how much increase in temperature, exposure to humidity can negatively impact the stability and usable life of the POLYOX product. Another way that we're finding that we can slow down the viscosity loss rate, slow down that auto oxidation rate, is through reducing the oxygen exposure to the powder. In this study, we cut the oxygen content within the package by less than half of the atmospheric oxygen. So atmosphere's oxygen is about 21%. We were able to get less than even half of that through using a new package type that one that allows for very, very slow oxygen migration through the packaging. Our current packaging using fiber drums has no oxygen control associated with it. We will start we will offer this package type, this extended stability packaging, next year. So look for that next year. But I wanted to give you a little glimpse into why we're so excited about this packaging, because when we use this, we significantly slow down the auto oxidation rate compared to the fiber drums. The fiber drums data of average of several batches is this red line, this red dotted line, and you can see the viscosity loss over time. This gray area is the theoretical zone of potentially these batches going outside the viscosity specification. This dark gray means definitely they're out of specification. Reason we have the bars is because each batch starts at a different starting viscosity, so you can't so they have a different percent viscosity retained where they're going to fall out of the specification. So you can see the red lines were for this 3.0. This is I believe this is a 3.0. No, this is coag. So this is there he goes, coagulant grade. It's in specification up to 6 months. At 9 months, maybe some of the batches were in spec and some were not. This is why we have the 6-month retest interval. For these packages in the blue, green, and kind of light green in the new extended stability packaging, you can see that we are well above the potential min spec, even out to 24 months or 2 years. So reducing oxygen and good storage conditions. Those can be key to extending the usable life of polyethylene oxide. Okay. Everything I just talked about was POLYOX powder in drums or in that new extended stability packaging type. A lot of times we get questions around, well, how does that translate to stability in a formulation? You have POLYOX, you compact it into a matrix, and you put it on stability. What happens? Well, we've tested this with a model formulation using theophylline. So we've taken these are theophylline with a blended with coagulant, a high molecular weight of 5 million Daltons, some Avicel, pH 102, a low mag stearate, put on stability in ambient storage conditions for 0, 6, 12, and 24 months. And we see, and then tested for dissolution at those time periods. And we see very good, almost identical release profiles when stored up to 2 years once it's in that compact. And that makes a lot of sense because you're reducing exposure to oxygen because oxygen can't migrate into that tablet very easily. And so the rate of auto oxidation is going to go down. We put those same tablets in accelerated storage conditions and measure dissolution at 0, 1, 3, and 6 months. And again, up to 3 months, we're seeing really good dissolution profiles. At 6 months, accelerated storage conditions, again, that's 75% relative humidity and 40 degrees Celsius. We're starting to see that we're getting a little bit faster release profile, indicating that there's potentially PEO degradation. I'm not too surprised with this because, again, those storage conditions are so harsh for PEO, and now we're just pushing the limit of the auto oxidation inside that tablet. The other thing I take away from this data is that accelerated 6-month accelerated storage condition is not indicative of, say, even a 2-year storage condition. You have to do a really good model to understand how accelerated storage is related to ambient storage because those conditions are so harsh to polyox. It's just better to go with ambient storage to understand the true stability nature of the formulation. That being said, accelerated storage can be very useful to push a formulation to understand if there are potentially any instability issues within a formulation. We use this to understand impact of binder selection in a POLYOX formulation. So here we took POLYOX 301, mixed it with theophylline, magsteria, and different binders. On this slide, I'm showing lactose and mannitol. Put those on accelerated stability for up to 3 months and looked at dissolution. And with lactose, we see this rapid increase in dissolution rate over that storage time, which is indicative of PEO molecular weight decreasing over time and allowing for faster drug release. And same thing, we saw very similar, but maybe not the same drastic effect as lactose, but we saw something similar with mannitol. So these reducing sugars might be negatively impacting the stability of PEO within a tablet. There are plenty of binders, though, where we don't see this happening. Basic calcium phosphate is one, microcrystalline cellulose, starch 1500. These are all great binders to use with polyethylene oxide in a formulation where we're seeing very good stability in the formulation. Okay, and this is my last slide. I like to end with reviewing stability recommendations when it comes to POLYOX polyethylene oxide. First is place drums in a cool and dry location. I can't reiterate enough how bad high temperatures, high humidity are to the powdered polymer in a fiber drum. Refrigeration will prolong the usable life of the material. If you have maybe, say, like research samples, if you could put those in refrigeration. Ideally, if you have your drums and a refrigeration source, you can put the drums in there. That would be amazing to help prolong that usable life of the material. We will have the new extended stability packaging available next year with a retest interval of 24 months. So please look out for that new packaging offering. Retesting. Retesting is pretty important for POLYOX. PEO, the viscosity is the main stability indicator, and this needs to be measured at the time of the retest date to make sure that it's still within range of the, if assuming viscosity is important to the end application, it's important to measure the viscosity at that retest date to see if it's still within the usable range. Formulation stability. We saw that formulations start because you get that compact, you start removing oxygen, you slow down the auto oxidation rate within a tablet. There are some considerations to keep in mind. So first, if you're, I always say, check your formulation for compatibility concerns, especially if you're using reducing sugars like lactose and mannitol. Check to see if you're going to have any issues upon stability. Running that accelerated study can can sometimes pinpoint if you're going to have issues when you look at early time points like 1 or 2 months or 3 months at accelerated conditions. uh, there, if you see issues with the lactose and mannitol, there are different ways to try to overcome those. One potential way is to incorporate more BHT into the system as a free radical scavenger to help slow down that auto oxidation rate. When adding BHT to the system, or even if sometimes we find that people are using the existing BHT levels in POLYOX for stability purposes, maybe the API stability or other stability purposes other than maybe stability for POLYOX PEO itself. But if BHT is important to their system, make sure that you take steps to check the BHT levels, especially if using process steps that might cause BHT to sublime or be removed from the system, because it can be easily volatilized through things like fluid bed. You might have to add BHT back to that. And with that, those recommendations, I would like to say thank you for joining me today, and I'm happy to take any questions that have come in. And I'll turn off my stop sharing.
All right, fantastic. Thank you so much, Elizabeth, for such a great presentation. So we're going to get started on the question and answer session, but before we begin, I would like to remind our audience how to submit questions. You can submit them by typing them in the Q&A box, which can be found directly below the video player. So Elizabeth, we've had a few come in since your presentation. So I'm going to start with this one, which says, what is the triggering pH range for auto oxidation of PEO?
Good question. We haven't pushed that to too many extremes. We've seen within most of the pH ranges that we test, typically are between. I'm trying to think of how far we've pushed it. 6 to 8, I think, is mainly where we've been operating. We haven't pushed it beyond that. We don't see too much difference, but we know POLYX can be unstable at really high pHs. So my guess would be really high pHs are not going to be conducive to POLYX. Now, it would be interesting to do a more formal study to better push those extremes and see how that affects the auto oxidation.
Great, thank you. And our next question here from our audience is there any correlation between polymer chain breaking during prolonged tablet storage versus change in porosity due to the breakage? How do the two relate?
Okay, so change in porosity within the tablet versus just the standard auto oxidation. So I don't think we haven't studied that directly, but if you're increasing porosity, I would guess that you're allowing more oxygen to then permeate into the tablet, and I would expect higher porosity, more oxygen permeating to the tablet to then increase that auto oxidation rate. Good question, though. That's a really good thing that we could look at to study.
Okay, fantastic. And the next question here for you says, what kind of assay do you run when you retest for auto oxidation?
So all of our oxidation data is coming from viscosity measurement. So our specification, our C of A, we have a viscosity either measured at 5, 2, or 1 weight%. We um, we continue to we run that at time zero and throughout the study to look at how is that viscosity changing over time, and that is going to be our indication of auto-oxidation is that viscosity loss rate. We can calculate that into a chain decision rate. That's just math that we've done, but from just how we measure it based off of solution viscosity.
Thank you. And our next question for you is, do most of your customers store Polyox in the fridge rather than RT?
um, I don't know what percentage breakdown that is. I would expect that it's very difficult for folks to store large drums of material in refrigeration. So I would expect that most people are storing at room temperature. I do know when in some cases our customers are storing at refrigeration because they know the benefits. They've understand and have seen the benefits of storing at refrigeration. It helps with their supply chain to be able to keep it longer and know that it's going to stay relatively stable. But for the most part, I have a feeling most people are storing at room temperature.
Thank you. And we got two questions that kind of came in back to back about the packaging. So I'll just combine them so you can give the answer. So, one just being, what is the type of new packaging? And the second question being, will the new packaging that will be introduced in 2025 be used for all Polyox grades and for all customers? Or will customers choosing this packaging pay more?
Okay, so I can't go into details about the packaging type right now. What I can say is that it is food safe, food contact safe. We know that it reduces oxygen permeation into inside, and then and I'm trying to think of what else I can say about it. It's smaller. It's a smaller packaging type than our standard 140-pound drum. So yes, we are going to offer it. Um, I believe, hopefully, I'm not misquoting. I believe we're offering it on every pharma grade that we offer, um, but it's additional packaging option. It's not going to replace the fiber drums. Plenty of folks like the fiber drums can manage the stability through refrigeration or supply chain issues, especially for the high molecular weight, which only have 6 months. This new packaging offering is for those where it's more challenging to for the supply chain of a 6 month retest interval product. So we're trying to offer something for them, and also offer a smaller packaging option for those that don't want such a large drum option.
Thank you very much. And the next question for you is, for the stability study in humidity and temperature, was that performed in packed form or open status?
Oh, open. Yes. So that humidity, we tried both this was literally a baggie. So if I the temperature humidity study, we put material in a bunch of little baggies. We had them both zipped closed and open, and um, we 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 found that we got similar results for the zip close. It took a little bit longer for everything to migrate in, but it was definitely more obvious when the packaging, those little baggies were open. So that data is all open baggies.
Thank you. And our next question is, are there any differences in Polyox performance in tablets formulated with water soluble versus water insoluble excipients?
Oh, good, good question. So we've looked at a mixture of both. We found that it seemed to be we had tested more excipients or binders and other excipients than just the ones that I shared. The lactose and the mannitol were the ones that showed the degradation effects, and we believe it's more to do with the reducing sugar properties of it, but we haven't really pushed that theory further.
Okay, great. And next question for you is one of the challenges with extrusion of polyox is the downstream milling process. Do you have any recommendations to make PEO extrudes more millable without cryo processes?
Oh, I always recommend cryo processing to help because it can get such a mess if you're doing standard milling processes. It's that thermoplastic nature that melting property just is not really conducive to those processes. Um, it's a good question. I know we've talked about in the past trying to come up with more realistic recommendations other than cryo milling. So I will take that question back and see what we can do to help answer it.
Fantastic, thank you. Our next question for you is how is the stability in oral films?
Oh, the civilian oral films. Oh, good question. I have a colleague that did some of that work. I will need to follow up with her. I can follow up with whoever asked that question because I believe she did some stability work on the thin films. Theoretically, it's higher surface area, and so I could imagine that it would be probably has possibly have increased stability issues, or you could lose viscosity possibly faster at that under storage conditions than say a compact, especially depending on how the thin film was made. Is it is it solution cast, or is it extruded? I can follow up with my colleagues to see if she has formal data quantifying it.
Great, thank you. And our next question for you is how are the other attributes, not viscosity, listed on the certificate of analysis affected by storage and storage conditions?
Yeah. So um, BHT, I talked about BHT can evolve over time, and our data suggests that this process happens. It's really slow, and it's much slower than the viscosity loss rate at a given storage condition. So we've seen batches go out of viscosity spec, um, and the BHT content is has moved a little bit, but is still well within the specification and fairly close to the starting BHT value. It takes a long time for that BHT to sublime off. Moisture, PO can pick up moisture, but it's very slow at your typical relative humidity levels. Once you get above 75% or 80% relative humidity, then PO will start rapidly picking up moisture. So avoiding high humidity environments um, will help keep that moisture level in the powder stable. Again, what our data shows is that at ambient storage conditions, refrigerated storage conditions, that moisture change over time is so minimal compared to the change in viscosity. Yeah, so in general, it's really viscosity that's the main quality indicator for polyox PEO.
Great, thank you. And our next question for you is what are some considerations to keep in mind when working with and handling polyox PEO?
Uh, okay, so many things. PEO is slippery and when wet, and gels when wet. So trying to avoid too much dust formation is good to keep the environment safe handling. When cleaning up, start with the dry sweep process to remove most of your excess PO, if either from the floor or from a tablet press. Follow up by a non-solvent. That non-solvent will help remove polyox, but not have it gel. And then hopefully gotten rid of almost 90%, 99% of your PEO where on whatever you're trying to clean, and then you can go through with like a water-based solvent to remove the last of it. You really don't want to start with water. You're just going to have a gel mess that is going to be impossible to clean up. Other things to keep in mind, the melting behavior of PEO. I've seen this a lot where overcompressed, we get crowning on a tablet because of that, that melting thermoplastic property of polyox. Same thing in a press. If you have little areas where powder can accumulate, you might get accumulation. It melts. It might even burn, depending on the type of process you're running. You really need to be observant to make sure that you're not creating nice little areas for accumulation or extrusion of PEO.
Okay, great. And I will ask you one final question before we wrap up for today. Just as a note to our audience, any questions that weren't asked or made it into the session will be answered offline, so don't fret. So the last question for you, Elizabeth, is how can one apply quality by design principles to formulations containing POLYOX PEO?
Yeah. So each formulation is going to be needed, or you need to be evaluated on a case by case basis. Um, what attribute is important to the formulation? That needs to be determined for each formulation. In many cases, viscosity is going to be one of the driving factors for performance of a formulation. You can test the hypothesis in many ways. A simple test is using the higher molecular weight or higher viscosity and lower viscosity grades from the grade that you're using in the formulation to see if that has an impact. That'll quickly tell you if you go too high in viscosity, you're going to be off on your performance metrics by so much. If you go low in viscosity, you'll be off, or maybe you're not off. And then that can help whittle down, oh, well, I need to look at increasing at how much increase in viscosity my formulation can handle. And that can be done by blending. You can take the grade that you're using plus, say, the higher viscosity grade, blend them to hit different viscosity rate targets and see how that impacts the formulation performance. Other properties that one may consider, I mentioned BHT. If that's important to stability of the formulation, you gotta figure out what levels of BHT you can operate within. You can easily do that by adding and subtracting or adding BHT, and I guess going on the lower side, you could always try to sublime the BHT. It's easier to add BHT than take it away. Let's see. Particle size might be another way. We've seen very little impact on particle size on the formulations that we've tested, unless you're talking sieving and using really high coarse particles and really fine particles. But usually within the particle distribution ranges of our grades, you don't see much impact on, say, a matrix tablet performance. But those are things that can be considered.
All right, fantastic. With that, we will wrap up today's event. I want to thank our audience for attending and for participating in today's event. I would also like to thank our sponsor, IFF, and Elizabeth for making today's educational webcast possible. As a reminder, if any questions weren't answered during this live session, they will be answered offline. We would like to ask everyone in the audience to participate in a brief survey. This survey will appear on your screen after today's presentation has ended. You'll receive an email alerting you, and this webcast will be available for replay. And we invite you to forward that announcement to your colleagues who may have missed today's live event. Thank you, everyone, for joining, and we'll see you next time. Have a great rest of your day.